How did they know that humans share 50% of our DNA with bananas? Well, I happen to know that there are several techniques scientists use to figure this out, and if you stick around till the end of the video, you will find out too. If we've never met before, my name is Nigel and I graduated with a 99.05 ATAR in 2023 and received high grades in biology and chemistry subjects, and it's my pleasure to bring you today's video on comparative genomics. So before we talk about the different ways to analyze DNA, we need to ask ourselves, what is DNA? Your DNA is the genetic code for your body. It is responsible for producing specific amino acids, which produce specific proteins, which produce specific tissues, which produce specific organs, and you put that all together and we get your body. Well, how does this whole process even start? Within the DNA, you can see that it has these strands holding it together. These are called base pairs, and there are 4 different bases you need to be aware of. Adenine, which binds to thymine, which is another base pair, and then cytosine and guanine, which bind to each other. And they arrange themselves in unique arrangements throughout the 3.1 billion different base pairs there are within the body. The types of amino acids that your body produces is dependent on the sequence of these base pairs, which can then influence the proteins made and the tissues made, etc. etc. Between humans, we share about 99% of our DNA with each other. It is the variations in that other 1% which can influence things such as your skin color, your eye color, your height, basically anything that makes you, you. So how can we compare our DNA with other species? Well, it's because DNA is universal. All living creatures rely on DNA in order to survive. Another thing which really helps is that no matter what species the DNA is in, it's always going to have those 4 specific base pairs that we talked about before, which makes an even fairer comparison. So now we need to talk about 3 different ways you can compare your DNA with other species. These are amino acid sequencing, DNA hybridization, and DNA sequencing. Like I said before, the sequence of bases in your DNA determines the amino acids which are made. So the more similar the sequence of amino acids are between different species, the more similar the DNA is and the more closely related the species are. So to do this, we need to use the same protein from each species, otherwise it wouldn't really be a fair comparison. That's like saying if I can kick a soccer ball further than you can kick a ping pong ball, then I'm a better kicker. Like, that doesn't make sense. No one said that. But the important thing is you gotta have a level playing field when you're making these comparisons. Although types of proteins vary between species, one protein which scientists love to use when doing these comparisons is cytochrome C, because cytochrome C is present in the mitochondria, and because every plant and animal has mitochondria, then every plant and animal has cytochrome C. It's such a fair comparison. So researchers can then observe the amino acid sequence in these proteins and then stake their claim regarding how similar these species are to each other. So now moving on from amino acid sequencing, let's have a talk about DNA hybridization. If you look carefully at the word hybridization, you can see that it has the word hybrid in it, which literally means a thing made by combining 2 different elements. If you haven't caught on, that means that we are going to be combining DNA from different species. This is done by heating the DNA to make sure that those strands which are holding it together are broken. Then we are going to mix those heated DNA from different species together and then allow them to cool, allowing complementary binding between the bases to occur and then forming that hybrid DNA in that one strand is from one species and then a different strand is from another species. So you're probably wondering, how does this actually help us find out how similar the DNA is? The hybrid DNA is then reheated to find the melting point. Essentially, the higher the melting point of the hybrid DNA, the more complementary bonding has occurred between the strands from different species and the more similar the 2 DNA sequences are. Let me give you an example. Let's say that each of my hands are a different strand of DNA. If we have 4 base pairs out of the 5 which are in common, they are going to bind to each other, and that's going to create an even stronger bond. So you're going to need more heat in order to break that apart and melt it. But let's say that there is only one complementary base pair. It would be a very, very weak link, and then you wouldn't need that much heat in order to break that bond. So yeah, basically the higher the melting point, the more similar the DNA of those 2 species are. Now let's move on to DNA sequencing. DNA sequencing literally means that you use some sort of machine, such as this Illumina next generation sequencing machine, in order to detect and map out the base pairs of the DNA that you are testing. Because this method gives you the exact base sequence of the DNA, you can literally just compare the DNA from different species and observe the differences in base pairs. This is very similar to what we talked about in amino acid sequencing. The more similar the base pairs are, the more closely related the different species are. So now that we have all this information about how similar different species are to each other, what can we actually do with it? Well, scientists believe that all organisms evolved from one superorganism called a LUCA, and we can map out where each species was created using a phylogenic tree. It's very similar to a family tree. Along the tree, you can see that as time goes on, more and more species begin to diverge from this LUCA organism, and then more species begin to diverge from those species, and so on and so on. If we find that two species have very similar DNA, we can assume that they haven't had much time to accumulate mutations and diverge even further. This means that they share a more recent common ancestor, which is that point in the phylogenic tree where the species diverge. I'm going to use my family tree to give an analogy on how this works. So me and my brother, we have very similar DNA. So I guess you could say that we diverged very recently from our parents. However, my cousins have very different DNA to what I have, which I guess you could say that they diverged a bit further up the family tree from where our grandparents were. But yeah, the more closely related two different species are, the more recently they diverged from a common ancestor. And there you have it. Now you know the basics of comparative genomics and how scientists use this to try and map out where different species diverge from each other. To stay updated, why don't you hit that subscribe button and hit that notification bell. Also, make sure you leave us a like and comment below on what lessons you want me to do next. Also, why don't you head over to our Instagram page and give us a follow there. This is where we post study tips and hacks for you to enjoy. Thank you so much for watching this video, and this is Nigel signing off.